Hello dear children, welcome back. I'm Dr. Jisha Joe. This is the second video on the chapter cell cycle and cell division. In this video, we shall study the different stages of mitosis. What is mitosis? Mitosis is a type of cell division that takes place in somatic cells. Somatic cells means body cells. So mitosis occurs in body cells. And mitosis is called as equational division. So first point to remember is that it is a type of cell division taking place in somatic cells. Somatic cells refer to body cells. The second point to be remembered is that mitosis is otherwise called equational division. Equational division. That is because the daughter cells produced are exact copies of the parental cell. The chromosome number of the daughter cells remains the same as that of the parental cell, and that is why mitosis is referred to as equational division. Can be asked is give reason. Mitosis is referred to as equational division. The reason is that the daughter cells produced have the same number of chromosomes as that of the parental cell. Another point to be remembered is that at the end of mitosis, two daughter cells will be produced. Two daughter cells are produced. And the peculiarity of the two daughter cells is that they are exact copies of the parents. They are exactly similar to the parental cell. Okay, so mitosis occurs in somatic cells. Mitosis is otherwise called as equational division because the daughter cells produced will have the same number of chromosomes as that of the parental cell. And at the end of mitosis, two daughter cells will be produced. Now we have already seen in the previous video, we have discussed about interphase. What is interphase? Interphase is the period of growth. We have studied that interphase is the period of growth. It is the period during which the cell prepares itself for the cell division phase. Now, what is happening during the interphase? We have also seen that the cell enlarges in size, cytoplasm increases, RNA and proteins increase, then the duplication of the DNA take place, centrioles duplicate, all these occur in the interphase stage. Now, we also saw that interphase is considered as a resting phase. Why is it called as a resting phase? Because the changes that are taking place in the chromosomes are not visible externally. So interphase is called as a resting phase. So during the interphase, the chromosomes appear as long, thin threads, a network of long, thin threads. That is how the chromosomes appear in the interphase stage. This network of long, thin fibers present in the interphase nucleus is called as the chromatin. Okay, now as the cell proceeds towards cell division, we have in the last video we have discussed the different stages of interphase. The final stage of interphase just prior to the cell division phase is the G2 phase, or the pre-mitotic gap. So what happens as the cell proceeds or enters into the cell division stage, cell division phase, is that these long network of fibers condense and coil to form the chromosomes. So if suppose a cell has two chromatin fibers, by the time the cell enters into the cell division phase, the number of chromosomes will be 2. What are chromosomes? Highly condensed, coiled chromatin fibers. Suppose a cell is having 10 chromatin fibers. How many chromosomes will it have? It will have 10 chromosomes because chromosomes are condensed, coiled chromatin fibers. So if we take the example of human cell, we know each and every cell of the human body has 46 chromosomes. So in the interphase stage, every cell will have 46 chromatin fibers. As the cell enters into the cell division phase, each of these 46 chromatin fibers condense and coil to form the chromosomes. Okay, and we have seen that each chromosome undergo duplication in the S phase of the interphase. So by the time the cell enters into the mitotic stage, each chromosome will have two arms, which are called as the chromatids, which are joined at a region called as the centromere. Now, I am going to draw a cell in the interphase stage. That is an animal cell that I am drawing at the interphase stage. So, animal cell has an outer membrane which is called as the cell membrane. During the interphase stage, the cell has an intact nucleus. Nuclear membrane is intact. It has the nucleolus and the nuclear material or the chromatin fibers are present there inside the nucleus. In the form of network of fibers, the chromatin fibers are present. So that is a cell at the interphase stage. That is an animal cell. We have seen during interphase, duplication of the chromatin or the chromosomes have taken place and the centrioles have duplicated, isn't it? Centrioles have duplicated. Okay, so that is that will be the structure of an animal cell at the interphase stage. That is an animal cell because it has an outer membrane which is the cell membrane. Cell membrane is present and it has the centrioles. Centrioles are present only in animal cell. Centrioles are absent in plant cell. Okay, and the nuclear membrane and the nucleolus, they are intact. Nuclear membrane. And that is the nucleolus. That is the chromatin. Okay, now this cell is entering into the mitotic stage or the cell division stage. Now, mitosis can be studied under 4 phases. Okay, the 4 phases of mitosis are: number 1, prophase; number 2, metaphase; number 3, anaphase; and number 4, telophase. So these are the 4 phases or the stages of mitotic cell division. Okay, this is a cell in the interphase stage. Now this cell is entering into the cell division phase, which is the mitotic phase, and the first stage of the first phase of mitosis is prophase. What happens during prophase? So by the time the cell enters into the first stage of mitosis, that is prophase, what happens? First thing is that the chromatin fibers condense and coil to form the chromosomes. That is the first thing to be remembered. The chromatin fibers in the interphase stage, the chromosomes were in the form of network of long threads. And by the time it enters into the prophase stage, these chromatin fibers condense and coil to form the chromosomes. Okay, and if you look at each chromosome will have 2 arms which are called as the chromatids, joined at a region which is called as the centromere. Okay, third point, the nuclear membrane and nucleolus start to disappear. By the end of prophase, nuclear membrane and nucleolus will completely disappear. The duplicated centrioles, the centrosome has duplicated during the S phase, and the duplicated centrioles will start moving to the opposite poles of the cell. This from the centrioles, the asters will be produced. Radiating rays, microtubular rays are called asters, which develop into the spindle fibers. So radiating rays called asters develop from the centrioles, and by the time the centrioles reach the opposite poles, they start to develop spindle fibers. So these are the things that are taking place during prophase stage. So first thing, chromatin fibers condense and coil to form the chromosome. Second point, each chromosome will have two arms which are called as the chromatids joined at the centromere. Third point, nuclear membrane and nucleolus disappear. Fourth point, the duplicated centrioles start moving to the opposite poles of the cell. They start to produce asters, which are radiating rays arising from the centrioles. So by the end of prophase, the centrioles will have reached the opposite poles. The nuclear membrane and nucleolus have completely disappeared. Centrioles will have started to produce the spindle fibers. Okay, so I'm just drawing the animal cell at the prophase stage. So that is the cell membrane. Okay, that is the cell membrane. We have seen the nuclear membrane is starting to disappear. That is why it is drawn in dotted line. That is the nucleolus. Nucleolus and nuclear membrane starts to disappear. Now I am taking an animal cell having just two chromosomes. Okay, so two chromosomes. How will the chromosomes appear? Each chromosome will have two arms. So that is the first chromosome I am drawing. And that is the second chromosome I am drawing. So if you enlarge the chromosome, if you just enlarge one chromosome, it will appear like this. It will have two arms. The two arms are called as the chromatids. Chromatids, they are the sister chromatids because they are the arms of the same chromosome. They are joined at the centromere. So each chromosome will appear like this. Okay, then what is the next point? The centrioles start moving to the opposite poles of the cell. They start to move to the opposite poles of the cell. Okay, I'm just labeling the parts. That is the cell membrane. That is the disappearing nuclear membrane. That is the disappearing nucleolus. Okay, that is the centriole. The duplicated centrioles are seen, they have started to move to the opposite poles. That is one there, second one. So they have started to move to the opposite poles. These are the chromosomes. Each chromosome will have two arms. That is because each of these chromosome has undergone duplication during the S phase of interphase. So this is how a cell will appear during the prophase stage. That is the prophase. So once again, prophase, what are the things to be remembered? The chromatin fibers condense and coil to form the chromosomes. Each chromosome will have two arms which are joined at the centromere. The two arms of a chromosome are called chromatids. Third point, nuclear membrane and nucleolus start to disappear. Fourth point, the duplicated centrioles start to move to the opposite poles of the cell. Asters begin to form from the centrioles. Okay. Now, by the end of prophase, after prophase, which is the next phase, metaphase. By the end of prophase, the cell, what might have happened? The nuclear membrane and the nucleolus have completely disappeared. The centrioles have moved to the opposite poles of the cell. So, I am just drawing the cell at the second stage, that is metaphase stage. That is the cell membrane. The nuclear membrane and nucleolus have completely disappeared. That is the centriole with the asters. The radiating rays around the centrioles, these are the asters. Radiating rays are called as the asters, that is the centriole. The centrioles have reached the opposite poles. They have started to produce the spindle fibers. Spindle fibers begin to form from the centrioles. Spindle fibers are microtubular fibers arising from the centrioles in animal cell. So, first thing, what happens? By the time the cell enters into metaphase, the nuclear membrane and nucleolus have completely disappeared. You cannot see the nuclear membrane and nucleolus there. The centrioles have reached the opposite poles and they have started to produce the spindle fibers. These spindle fibers get attached to the centromere of each chromosome, and during the metaphase stage, you can see the chromosomes at the equator of the cell. At the equatorial plane of the cell, you can see the chromosomes. So that these are the two chromosomes there lying at the equator of the cell. Okay. Sorry. So, spindle fibers get attached to the centromere and the chromosomes get pulled or arranged at the equatorial plane of the cell to form the metaphase plate. So, that is the metaphase plate. So, if these are the poles, this will be the equator. If for a particular cell, if these two are the poles, then the equator will be in this plane. So, what is to be remembered during metaphase? The chromosomes get arranged at the equatorial plane of the cell. How is this happening? The spindle fibers arising from the centrioles get attached to the centromere of the chromosomes, and the chromosomes get aligned at the equator of the cell, forming the metaphase plate. Okay, so that is the metaphase plate formation during the metaphase stage. So that is how we identify a cell at the metaphase stage. How? Look at the chromosomes. If the chromosomes are arranged at the equator of the cell, then that is the metaphase stage. And it is during the metaphase the chromosomes are in the most condensed state. And it is during the metaphase stage the study of chromosomes is done. Because chromosomes are in the highly condensed stage and we are able to identify each and every chromosome. So, study of chromosome or karyotyping is also done during the metaphase stage. So, that is with regard to the metaphase stage of mitosis. So, labeling is not complete. They are the centrioles. That is the centriole there. You can label the spindle fibers. Spindle fibers, the chromosomes. These are the chromosomes. The centromere, all these can be labeled there. Okay, so that is the metaphase stage. Now moving on to the third stage. So metaphase. Now moving on to the third stage of mitosis. Which is the third stage of mitosis? It is anaphase. What happens during anaphase? During anaphase, the centromere of each chromosome splits. The centromere splits and the sister chromatids are pulled to the two opposite poles of the cell by the contraction of the spindle fibers. So I'm just drawing an animal cell, the same cell at the anaphase stage. It has the cell membrane there. Then it has the centrioles at the opposite poles. One centriole, the second centrosome there. Then what happens during the anaphase? During the anaphase stage, the centromere of each chromosome splits. Where are the spindle fibers attached? The spindle fibers are attached to the centromere. Now, so since the centromere splits, when you draw, remember the centromere should be directed towards the poles because the spindle fibers are attached to the centromere. Okay, so that is the way you have to draw the chromosomes, because pulling of the chromosomes take place here. So centromere should be directed towards the poles. So here what has happened? The centromere has split and the sister chromatids are pulled to the opposite poles of the cell. How? By the contraction of the spindle fibers. The spindle fibers contract and the sister chromatids are pulled to the opposite poles of the cell. So these are the sister chromatids. Okay, so that is the cell at the anaphase stage. Okay, so what is to be remembered during anaphase stage? The centromere splits and the sister chromatids are pulled to the two opposite poles of the cell. How? By the contraction of the spindle fibers. Where exactly are these spindle fibers attached? The spindle fibers, I told you, that are attached to the centromere. The exact attachment of the spindle fibers is to the kinetochores. What are kinetochores? Kinetochores are disc-shaped structures present at the centromere. Okay, so spindle fibers are attached to the kinetochores of the centromere. In one of the previous videos, we have discussed about the centromere, which is a primary constriction. We have discussed about the kinetochores, etc. So please go through that video. If you have a doubt, please refer to that video. So that is anaphase. Okay, so spindle fibers are attached to the disc-shaped kinetochores of the centromere. So we have completed anaphase. Now the final stage of mitosis is the telophase. Telophase is just the reverse of prophase. Why is telophase called as the reverse stage of prophase? Because whatever has happened during the prophase, just the reverse of it takes place during the telophase stage. So what has happened during the prophase? We have seen the first point, the chromatin fibers condense and coil to form the chromosomes. In telophase, what happens? The chromosomes decondense or uncoil to form the chromatin fibers. Cell division, that is telophase, is the end of mitotic phase. So now the chromosomes, the condensed chromosomes decondense or uncoil to form the chromatin fibers. The second thing that we have seen is the disappearance of the nuclear membrane and the nucleolus. So here what happens? The nuclear membrane and the nucleolus reappear. The third one, the spindle fibers disappear because now the function of the spindle fibers is over. The spindle fibers disappear. Okay, so by the end of anaphase or by the start of telophase, the sister chromatids have reached the opposite poles of the cell. and these sister chromatids decondense or uncoil to form the chromatin fibers. Spindle fibers disappear because their function is over. Nuclear membrane and nucleolus reappear. Okay, so that is what is happening during the telophase stage. So the decondensation of the chromatin, a decondensation of the chromosomes to form the chromatin, the nuclear membrane and nucleolus reappeared. Okay, and the centrioles are present there close to the nucleus. The centrioles will be present. The spindle fibers have disappeared. Now that is the end of mitosis. That is the end of nuclear division, which we have studied in the previous video. Mitosis is divided into two stages: karyokinesis and cytokinesis. Karyokinesis is the division of the nucleus. Nuclear division, karyokinesis, means the division of the nucleus. So nuclear division is complete. Now the cell is entering into the second stage of mitosis, that is cytokinesis, which is the division of cytoplasm. So in animal cell, cytokinesis occurs from the periphery. Furrows form, or furrows start to develop from the periphery, and they extend to the center. So in animal cell, cytokinesis occurs by the formation of furrows. You can see furrow formation taking place from the periphery, which extends towards the center. So this type of cytokinesis, or the direction of cytokinesis here in animal cell, is centripetal. That is, furrow formation occurs at the periphery and it extends towards the center. So extension occurs like that. Okay, the furrow extends towards the center, and finally the two join together and two new cells are produced. So that is the telophase stage. Telophase. Okay, so remember, in animal cell, how does cytokinesis take place? By the formation of furrows. Where are the furrows formed? The furrows are formed at the periphery, at the cell membrane. The cell membrane forms the furrows. The furrows extend towards the center, and hence the direction of formation is centripetal. Okay, centripetal. Whereas in the case of plant cell, that is in the case of animal cell, whereas in the case of plant cell, suppose if that is a plant cell, so plant cell, we know intact cell wall is present. It will have an inner membrane which is called the cell membrane. Okay, all the other changes or different phases are the same, but here the wall formation or cytokinesis, or the division of the cytoplasm, is different in both plant cell and animal cell. In plant cell, cell plate formation takes place from the center and extends towards the periphery. As you can see in the diagram, the direction of the arrow shows the extension of formation of the or the direction of the formation of cell plate. So in plant cell plate formation takes place from the center towards the periphery, and the cell plate represents the middle lamella, and later on it is on to this middle lamella. Middle lamella is rich in calcium and magnesium pectates, and it is in one of the previous videos, while studying the structure of the cell wall, I have discussed about the middle lamella and all. So later on onto this middle lamella, the primary wall and the secondary wall gets deposited. So in plant cell plate formation takes place at the center and extends towards the periphery. So here the direction of growth of cell plate or the wall formation occurs in the direction is centrifugal. Whereas in the case of animal cell, we have seen furrows are formed at the periphery, and these furrows extend towards the center, and here we can say it is centripetal. In some cases, karyokinesis will not be followed by cytokinesis. In such cases, when karyokinesis is not followed by cytokinesis, what will happen? Within the same cytoplasm, there will be two nuclei. So here, karyokinesis is followed by cytokinesis. That is why the nuclei are getting separated there. If cytokinesis does not occur, what will happen? Within the same cytoplasm, many nuclei will be present. Such a condition is said to be syncytium. Syncytium. That is a multinucleate condition. And the formation of syncytium is due to the absence of cytokinesis after karyokinesis. Now we are moving on to the last part of this session, the significance of mitosis. What is the significance of mitosis? The first important significance is that mitosis is necessary to maintain the chromosome number. We have seen at the beginning of the session that as a result of mitosis, the chromosome number is maintained. The chromosome number of the daughter cells remain the same as that of the parental cell. The second important significance is that mitosis helps in the growth of body parts. The third important significance is that damaged parts need to be repaired. So cell division taking place for the repairing of the damaged parts occur by mitosis. Mitosis is the type of cell division taking place for the repair of body parts. Another important significance is that mitosis is necessary for the replacement of cells. Worn out tissue or worn out cells have to be replaced by new ones. This is again done by mitotic cell division. Now in the case of asexual reproduction, as in the case of bacteria, amoeba, yeast, etc., unicellular organisms, again their cell division takes place there. The cell division is a form of reproduction there. So the type of division taking place in such organisms is also mitosis. In plant cell, the increase in length of the plant or increase in girth or width of the plant again occurs as a result of mitosis. So my dear children, in this video we have studied the different phases of mitosis. We have also discussed the significance of mitosis. I hope whatever we have studied in this session is clear to you. If you have any doubts regarding what we have studied, please ask your doubts and also give your suggestions in the comment section. Thank you.